it's not often in your career you end up in science. So I congratulate Pat and his colleagues at Columbia on this work, and look forward to hearing what they have to say about the new virus. Pat Moore.
Thank you, Steve. Could I go ahead and have the first slide, please? Thank you. And I'll dim the lights a little bit, please. As Steve mentioned, we've recently published a paper in Science that has provided some evidence that herpes virus-like DNA sequences can be found in Kaposi sarcoma lesions. However, there are two major questions that we have left unanswered with this data that I would like to address in this talk today using some of our unpublished data. And that is, one, is there truly a new human herpes virus in Kaposi sarcoma lesions? And two, if this virus is in Kaposi sarcoma lesions, is it causal for KS? Now, with any study like this, you can imagine there are a lot of people who have contributed to this. I would like to mention that Yuan Chang, my colleague, lab co-director and occasional dinner mate, has been responsible for many of the experiments that I'll be showing here today, and has either directly performed them or has supervised them. And also Ethel Cesarman, who is now at Cornell, has been tremendously helpful and responsible for collecting together a large batch of tissues for the studies I'll be talking about. In addition, Melissa Pess and Dan Knowles, frankly, and Janice Culpepper have also been instrumental in the initial studies that I'll be talking about. Can I have the next slide? Oh, I guess I'd do that myself. In 1990, Harold Jaffe and Valerie Burrell published a paper in Lancet which, based on epidemiologic evidence, strongly suggested that Kaposi sarcoma may be due to a sexually transmitted exogenous infectious agent. And this paper certainly piqued our interest in this idea, and we decided to see what we could do to try and find a KS agent. We took the strategy of trying to isolate unique DNA sequences that are found in KS lesions that are absent from normal tissues, or at least uninvolved tissues from the same patient. We would identify these unique DNA sequences by using a subtractive hybridization and kinetic enrichment technique called representational difference analysis, or RDA. And then our idea was to, once we'd obtained these unique DNA sequences, test them using southern hybridization, which is a very specific test for binding to Kaposi sarcoma DNA, and also design PCR primers, which is a very sensitive test for detecting unique DNA sequences. We performed RDA on an AIDS patient who had KS who came to autopsy, and compared his Kaposi's DNA to uninvolved DNA. And after 3 rounds of RDA, 4 distinct bands formed, which are shown on this slide. The 2 middle BAMs, KS390 BAM and KS480 BAM, very early on we realized that these were non-specific for KS. They're probably human DNA sequences, and we haven't pursued them further. But after an initial evaluation, KS330 BAM and KS631 BAM, the numbers representing the number of bases in the sequences, appeared to have some specificity for Kaposi sarcoma DNA. This is a figure from our paper. It shows that in our initial testing of a series of tissues using southern hybridization to the KS330 BAM sequence and KS631 BAM sequence, when we tested DNA that was extracted from 27 AIDS KS lesions, 20 and 21 of them hybridized to these 2 bands. In addition, we sequenced the KS330 BAM sequence and designed unique PCR primers that amplify an internal fragment that's 233 base pairs long. And we tested these same 27 tissues with this PCR primer set and found that 25 of these tissues were positive for this PCR product. The 2 tissues that were negative were very interesting. One, when we went back to look at the tissue, it turned out not to be Kaposi's at all. The tube had been mislabeled. And the other sample probably had degraded DNA because it was not amplifiable for other PCR primers that give you products from human genes such as P53, a portion of P53. So these two samples that were negative appear to be negative for technical reasons. Now, we've tested 39 other tissues from AIDS patients that did not have KS in the tissue itself. 6 of these tissues are positive, 3 of which are a non-Hodgkin's AIDS lymphoma that I'll talk a little bit more about in a few minutes. And as you can see also that 25% of the AIDS lymph nodes that we tested were positive for these DNA sequences by all 3 assays that we used. In contrast, we tested 85 tissues from non-AIDS patients, which included 29 non-AIDS lymphomas, 7 non-AIDS lymph nodes, a panel of 5 vascular tumors, and 49 consecutive surgical biopsies, which were biopsies from tissues throughout the body. And we were unable to detect any of these sequences by these 3 assays in these patients. And then finally, we tested a series of tissues from both AIDS patients and non-AIDS patients that contained opportunistic infections like EBV, CMV, and PCP that are common to AIDS patients. And again, there was no reaction in any of these 3 assays. This is another diagram from our paper where we did at least a first approximation of tissue distribution of these DNA sequences. And I hope that you can read this in the back. If you can't, the first line under the patient is the KS lesion. These are 4 additional KS lesions above the 27 that we tested before. And all 4 KS lesions from these patients were positive by all 3 techniques. In patient A, both skin and muscle were positive by all 3 techniques, also suggesting dissemination of these sequences. However, in the remaining patients, the majority of tissues were negative by all 3 techniques from a wide variety of tissues. The exception to this is stomach from patient C, which is adjacent to the KS lesion here, that was positive only by PCR, and then skin, which was adjacent to the KS lesion in patient D, as well as hilar lymph node from patient D, which were positive by PCR. Again, this suggests that these sequences are present in these tissues, but at a threshold pulled below the level of detection by southern hybridization. Now, this kind of pattern strongly indicates that this is not a human polymorphism that codes for susceptibility to KS, of course, and it is the pattern that one would expect for an exogenous infectious agent. But I'd also like to point out one more thing, is that at least in a crude fashion, we now have something of a biological gradient that we're starting to see with these tissues, in that tissues that are more proximal to the KS lesion are more likely to contain these DNA sequences than more distal sites. Well, if this is an infectious agent, then what is it? We've used the KS330 BAM sequence to screen a genomic library made from a KS lesion. We've pulled out a 21 KB, that's not quite in focus, is it? A 21 KB lambda-phage clone that hybridizes to the KS330 BAM sequence. I'm sorry, this slide is obsolete now. This was made about 3 weeks ago. And we've completed the sequencing of this lambda-phage. We have about 95% confirmatory sequencing on this lambda-phage, and so now we're fairly confident of the sequence. We've identified 15 different partial and complete open reading frames on this lambda-phage clone. This schematic diagram on the bottom is the corresponding region of the genome of a virus known as herpes virus thymeri, which is a gamma herpes virus. And we've identified 14 of 15 open reading frames, which are in the same position and have high levels of homology to these corresponding herpes virus thymeri genes. Now, herpes virus thymeri is a gamma herpes virus that is found in New World monkeys, and it can cause a fulminant lymphoma in some species of New World monkeys. There's only one open reading frame right here at what corresponds to the homologue for ORF28 in herpes virus thymeri that shows no homology to herpes viral genes. This is right at a break point between 2 conserved gene blocks in gamma herpes viruses, and so it's not too unexpected that we would lose homology at this point. Some of the important genes that we have on this include a homolog that has reasonable homology to the thymidine kinase gene of herpes virus Cymeri, the GH glycoprotein, and the major capsid protein, which is a major component of the viral capsid, as well as VP23, which is a minor capsid protein. And the VP23 gene is where the location of the KS330 BAM sequence is, right here. In addition, something that's not marked on here, there is a putative DNA packaging protein, which is a splice gene that is highly conserved. It's composed of these two sequences here. Now, we haven't done a formal alignment because we are still sequencing and confirming the sequence, but we've at least performed an alignment through BlastX, and that's shown on the side here, where we have the approximate amino acid identity for the hypothetical proteins made by these open reading frames compared to the similar proteins that are the homologous proteins that are made by herpes virus Cymeri. And you can see that at least two of these open reading frames, the open reading frame for major capsid protein and for the putative DNA packaging protein, have greater than 75% amino acid identity on this first analysis with corresponding proteins in herpes virus Cymeri. Based on this amount of sequencing and also the next set of slides that I'll be showing you in a few minutes, we, for convenience sake, are referring to this, which we believe is a new virus, as Kaposi sarcoma-associated herpes virus. This is a temporary name and it's for convenience only. There are formal names for herpes viruses, and we do anticipate that the name will change over time, but we prefer to abbreviate it as KSHV until there is a formal designation. Additional evidence that this is a gamma herpes virus comes from the splice gene region. Here's exon 1 and exon 2, and this region has been used by other authors to place the phylogeny of other human herpes viruses such as HHV6. You can see the nucleotide sequence in this region and also the translated amino acid sequences for these regions are similar, most similar to the corresponding gamma herpes viral sequences. This is herpes virus Cymeri here, and Epstein-Barr virus, which is another gamma herpes virus, which has fairly high levels of homology to the sequences that we pulled out of the genomic library. Now, 3 of the 27 AIDS lymphomas were positive in our initial screening of a panel of AIDS tissues. And co-authors. Ethel Sezerman remembered that several people, including Gianluca Gaetano and Ricardo della Favera, and then secondly, Giorgio Ingarami and Dan Knowles, had separately placed cells from these lymphomas into culture, and knowing that these lymphomas were positive, we raced to the refrigerator, thawed them out, put them in culture, and tested them with our by southern hybridization and by PCR. And indeed, these cells in continuous in vitro culture are positive for these DNA sequences, and sequencing of, for instance, a PCR product from these cells shows that they are virtually identical to the sequences that we pulled out of our genomic library. The cell line is EBV co-infected. It's probably an EBV producer cell line, and it periodically undergoes a cytopathic effect that we're still trying to characterize. Yuan Chang has taken the cells and performed a pulse-filled gel electrophoresis to get an idea of the size of the genome, and this is a picture of our gel that's been hybridized to the KS631 BAM sequence. You can see that there's a band here that is probably episomal DNA, which would be characteristic for a herpes virus. That's located at about 270 kb. Now, this is an extremely large genome for a herpes virus, and it's certainly the largest human herpes virus genome, and we have repeated this several times, and I think that this is is the real size of it, but of course this is probably a circular molecule, and its migration on the gel may be a little bit different than this when it's linearized, but it's probably nonetheless a very large genome. Rao Shiganti at Sloan Kettering Memorial has been kind enough to do fluorescent in situ hybridizations for us on the cell line. The panel on the left is an interphase spread for the cell line, which we call BCBL1, and you can see there are multiple hybridizations in this nuclei, indicating that there are multiple copies of the virus in the cell line. On the metaphase spread here, I can't really say this, but Rao tells us that this is indicative, this pattern is indicative of an episomal DNA that's physically aggregating with the chromatin. Certainly there's not any clear single insertion site for the DNA into the host chromatin, and so it's likely that this is showing us that indeed the DNA is episomal, which again would be characteristic for a new human herpes virus. We've performed some preliminary pelleting experiments where we took a small amount of supernatant, performed low-speed centrifugation to remove cells, and intermediate-speed centrifugation and filtration, and then repeated it again to remove nuclei and other organelles, and then we pelleted the supernatant for 100,000 G for 1 hour. And we can get PCR pellet from this washed, a PCR product from this washed pellet. This is a KS330 sub 233 PCR signal right here. We also have performed PCR using EBER-specific primers, and we're pelleting, co-pelleting the Epstein-Barr virus genome at the same time. Is it possible to turn out the lights? I don't know that you can actually see this very well. Well, if you can turn them down a little bit, otherwise I'll try and tell you what's on the slide. We've also done some preliminary experiments to form a serologic link between the cell line and KS patients. This is an indirect immunofluorescent assay on the side. This side here is a photomicrograph of the cells, and on this side, perhaps you can see that you'll have to take my word for it, that cells are staining with antibody here. This is a control sera from a patient who had AIDS. It was a woman who had AIDS but did not have KS at autopsy. Next slide, please. Oh, sorry. And then after we adsorb her sera with paraformaldehyde-fixed Raji cells, we're able to adsorb out specific antibody binding to the cell line. In contrast with a KS case here, this is the KS case after adsorption of the patient's sera with the Raji cell preparation to remove anti-lymphocyte and perhaps anti-EBV antibodies, we still see specific antibody binding to the cell line. And this is summarized in this slide, and if you want to turn up the lights again, I think we're past the bad slides. This is data from sera that Isaac Weisfuss at the New York City Health Department kindly provided us. We had 4 patients who had KS and came to autopsy in a study that he was working on, and 4 control patients with AIDS, all of these were AIDS patients, and 4 control patients with AIDS prior to absorption. Both the case patients and the control patients have relatively high IFA endpoint titers to the cell line, but after absorption, the control titers tend to drop off remarkably, whereas the case titers remain high. And so I think that this is at least preliminary evidence suggesting that there may be a specific humoral response directed against this cell line in the KS patients, which again would be what one would expect if this herpes virus is associated with KS and if it's present in the cell line. We've repeated this about 3 times on blind reads, and so I'm beginning to believe that this is real data now. In addition, I'd like to talk a little bit about KS in HIV-negative persons. We sent them to us in a blinded case control evaluation We had 21 cases that we evaluated. 11 of them were AIDS patient DNAs, that was DNA from KS lesions from AIDS patients. We had 6 DNA samples from classical KS lesions, and then we had 4 DNA samples from HIV-negative gay men with KS from their KS lesions. And as you may know, there's at least some evidence that suggests that HIV-negative gay men are at somewhat higher risk for developing KS than the general HIV-negative population. For a control group, we used peripheral blood monocytes from 10 different donors and 11 excess plastic surgery skin samples, again from 11 separate donors. We performed PCR on these samples and did confirmatory southern blotting against the PCR product to ensure that we were getting specific amplification. 20 of the 21 KS samples were positive. Only 1 of the control samples was positive, giving us an odds ratio of 400 with confidence intervals clearly excluding 1. The cases that were positive included 10 of the 11 AIDS cases. There was 1 case of AIDS KS where we received multiple blind samples of his DNA, and it was uniformly negative by PCR for these sequences. All 6 of these classical KS, HIV-negative classical KS DNA samples were positive, as were the 4 HIV-negative KS samples from gay men. In the control group, there was a single skin sample that was positive on our initial evaluation. When we repeated the evaluation of it, it was generally negative. However, we've kept this in for as a conservative assumption as being positive. I think that this is probably a false positive result of our PCR. We've sequenced representative PCR products from 3 AIDS KS tissues, from 2 classical KS tissues, and from 2 HIV-negative gay men who had KS from their tissues. The products were TA cloned and sequenced, and we found 2 distinct genotypes among these 7 PCR products that differed at 1 base pair position or more from the prototype sequence derived from our genomic library. But overall, these sequences had greater than 98% nucleotide identity with the genomic library sequence, suggesting that they are the same virus. We did not notice any correlation between the genotype of the PCR products and the type of KS. And finally, we've been working with John Ziegler, Edwin Katangoli Mabiri, and Henry Wabinga at the Uganda KS project. They're in Kampala, Uganda. And as many of you know, KS can occur in parts of Africa at a very high rate in HIV-negative individuals, where it's called endemic African KS. This group has sent us 21 paraffin-embedded KS specimens, including 12 AIDS KS specimens, which included 3 specimens from children under the age of 7 and 9 HIV-negative endemic KS specimens, 5 of which were from children under the age of 14. In the United States, KS is vanishingly rare if it occurs at all in children, but in Africa, endemic KS can occur in children where it is a fulminant illness and often rapidly fatal. This is a sample of our preliminary PCR results showing, this is just 10 of these samples, showing that all 10 of these samples do apparently amplify for our PCR product. And so this is at least strong preliminary evidence that the virus is present in endemic KS specimens, both from HIV-negative and HIV-positive persons in Africa. So I'd like to close by saying that I think that the evidence is pretty strong now that we actually do have a new human herpes virus in KS lesions. We see a collinear homology, and again, this is a bit out of date, over a 21 kb lambda-phage clone that we pulled out of a genomic library We've seen collinear homology at 14 of 15 different herpes viral genes on this lambda-phage clone. In addition, the KS631 sequence itself is homologous to the herpes virus Cymeri tegument protein gene, so we have homology in two physically separate regions. Some of these open reading frames have extremely high amino acid identity to known herpes virus Cymeri proteins, including the major capsid protein and the DNA packaging protein. The virus appears to be in continuous in vitro culture. We've performed some preliminary experiments to demonstrate that it can be the DNA sequences can be filtered and pelleted, and we at least have some evidence of a serologic link between the cell line and Kaposi sarcoma. Without coming right out and saying that this is the cause of KS, I think that we're at least heading in that direction. We see that there's a very strong association between the presence of these DNA sequences and KS, that these sequences tend to be highly specific for KS. They are also found in this non-Hodgkin's lymphoma that occurs in AIDS patients. I won't talk very much more about that because Ethel is taking the lead on exploring that, and she's preparing that for publication. But aside from that disease entity, we generally don't see it in other tissues from other types of diseases, at least among the tissues that we've tested so far. The findings appear to be very generalizable. It appears that the DNA sequences are present in all forms of KS that we've tested so far. There is at least some evidence of a biological gradient in that tissues that are near proximal to the KS lesion are more likely to be positive for the sequence than more distal sites. There's a great deal of biological plausibility that this herpes virus is the cause of KS, in that related viruses such as Epstein-Barr virus and herpes virus Cymeri are both tumorigenic. I believe this is reproducible. We've heard at least rumors of numerous labs being able to reproduce our PCR results, and so I think it does work. And then finally, Larry Kingsley has been very, very helpful and Chuck Rinaldo have been very helpful in providing us with some tissues for confirmatory studies. We haven't had a chance to show them here, as have Harold Jaffe and Scott Holmberg at CDC. But I would like to say that we've been doing some work with lymphocytes from Larry Kingsley's group. We have some lymphocytes that were taken prior to the diagnosis of a patient with KS, and it looks like some of these lymphocyte samples are positive, although the data is not quite mature enough to present here. I think that it's likely that we will be able to demonstrate the correct temporal association, if not with PCR, then eventually with a serologic assay showing that patients are infected with this virus prior to their development of KS. So on that note, I think I'll close and open it up for questions.